Hello everyone, welcome back to another YouTube video. Today we're going to do the 2015 USABO Open Exam. I'm so sorry I haven't been able to post. It's been really busy with school and preparing for USABO itself. So yeah, like cause scores come out not today, not tomorrow, but the day after on the 28th. So good luck to everyone who is participating in that. So yeah, let's just get started with this exam. So like with the 2017 exam, I'm gonna do the first like 12 or 13 questions and then do 12 or 13 questions at a time till we get to 50. So yeah, we'll see how it plays out with everything. And yeah, let's just get started. All right, so our first question is, what best describes a nucleosome? A region in bacterial world where DNA is located. Well, and then we have all of these options. Essentially, you really need to know what a nucleosome is, and this is not something that really requires too much critical thinking. You either know it or you don't. It's kind of a unit of like a chromosome. So what happens is that you have like these histone proteins. Like there's more than this, but they're like the DNA is like kind of wrapped around it, and like this one unit where it's like wrapped around. And there's like multiple of these, but like this one unit is called the nucleosome. So let's see which one fits best. So region in bacterial cells. No, we have these as well. I don't think bacteria even have these, so A is not correct. And DNA wrapped around 8 histone cores, which is correct. I remember it does have 8, but even if you didn't, this one seems to be the best option for right now. I'll just put a check right next to it. RNA with protein bound. Nope, it's DNA, not RNA. A region in the nucleus where ribosomes are assembled. Nope, that isn't correct either. That is the where is it? The nucleolus. And then the space between the layers of the nuclear membrane. No. So clearly the answer is B. That's the one that is remotely close to what I said. So B would be the answer. All right. Let's go to question number 2. You observe fly cells that are undergoing meiosis and treat them with a drug that inhibits the separation of parental chromosomes. Okay, this is a very common technique with the USABO. Whenever you see that something is inhibiting or like a drug is inhibiting a certain process, that's kind of like what you need to focus on. So because it's inhibiting separation of parental chromosomes, I'm already thinking this is going to be meiosis II, because we already have like we have like the so we had like chromosomes, and now crossing over and all that stuff has happened, and meiosis I's happened. We have these now, and these chromosomes, these parental homologs, or the homologous chromosomes, are gonna be separated into each of their cells, or into each of the gametes, should I say. And then at which state of meiosis do cells treated with this drug become arrested? So first of all, I already know this it's not going to be any of these, because we're talking about parental homologs being separated. So it's not going to be meiosis I. Now the trick here, and this is a very tricky question, you already know telophase and prophase won't be the answer. The first like thing you think of is to go to anaphase, because that's when so we have this and metaphase, they line up in the middle, and then in anaphase, so I'm gonna write M here, and then in anaphase they get separated to the ends. So this would be anaphase. Wait, let me just draw that cell. This is a little bit weird. Where they got like separated to the ends. But the answer would actually be metaphase, because that like process of separation does start in metaphase. So if this is preventing the separation, it's going to first stop the metaphase from happening, and then which is going to prevent like that anaphase process from occurring. So that's why the answer would be AB. This one was a little bit of a tricky question, which required a little bit of deeper thinking within meiosis. So yeah, this was a pretty difficult question. All right, next question. Let's look at number 3. We have psoriasis is a skin disorder that is caused by the increased proliferation of skin cells and results in the red itchy skin cells from a mouse model for this disease are isolated and cultured in a plate containing a drug that promotes the supercoiling of DNA. Already, we have that drug thing again. I'm just going to underline it because the question is definitely going to be talking about this drug. And then you can look at the next question, which enzyme does this drug inhibit? Well, you can already tell C is bolded, so it is C, topoisomerase. And you really should know this enzyme though. It's very important in DNA replication. So, you know, when you have helicase, like, you know, breaking open everything, you then have like topoisomerase at the like other side of the replication fork. So let me just make a replication fork, right? Or that's not that good. Let's maybe have right here. Or, you know, that replication bubble. This topoisomerase is going to be right here to make sure that your DNA doesn't supercoil. And then you can have like DNA polymerase just go along here and, you know, just add the nucleotides. But yeah, topoisomerase is a very major enzyme. And okay. So then the next question is which which phase of the cell cycle do cells treated with the drug specified in question 3 become arrested? All right. So topoisomerase is the most active during, you know, DNA replication. And we know G0, that's not DNA replication. G0 is like where most nerve cells are because they don't divide. And then G1 and G2 are the two growth phases. The answer here would actually be S phase because this is where the DNA does get replicated in preparation for the next mitosis. So, and of course it wouldn't be M phase because M phase is mitosis, and that's not when it's replicated, but in fact when the chromosomes are divided. So that's kind of the opposite. So you already know it's not going to be these. So the answer would be E, S phase. So it's important to keep that in mind, knowing your mitotic cycles, and knowing mitosis in general is also very important. Mitosis, meiosis. All right. So we have a liquid culture of yeast was grown under anaerobic conditions, but then had a significant quantity of alcohol dehydrogenase inhibitor added to the medium. All right. Again, we have this inhibitor thing coming up, you know, drug inhibiting enzyme inhibition. So I'm just going to underline it just so that I can keep my eye on this. Which of the following would best describe the effect of glycolysis due to the presence of the inhibitor? All right. So we're in anaerobic conditions. So that means that fermentation is occurring. Now, there's two ways to tell which type of fermentation is occurring. This is not extremely essential, but first of all, yeast, they release alcohol in when bread is being made, for example, and then alcohol dehydrogenase, you can even tell from the name. So this is alcohol fermentation. And if we're having like alcohol dehydrogenase being inhibited, that means the alcohol is gonna unable to be like broken down, and like that process can't continue. So that means that we're gonna have excess like pyruvate from like glycolysis. So if I just make it like this is a very, very simplified version, but you have glucose, which becomes pyruvate, and that's gonna become what ethanol. Well, I think there is like a compound in between. That's important to know, but this is really the basics of alcohol fermentation. So if we're gonna have this unable to be broken down, we're gonna have too much of pyruvate. And that means that glucose isn't going to be broken down. So that means the answer would be E, because pyruvate is inhibiting the glycolysis, maybe not directly, but there's some sort of feedback inhibition going on, which would be like pyruvate inhibiting glycolysis. It would eventually lead to a stop because the ethanol cannot be like this alkyl dehydrogenase is inhibited. So yeah, lowering lactic acid and alcoholic fermentation is pretty important, as well as the cell cycle, especially like the major compounds and enzymes. So yeah, just knowing like glucose becomes pyruvate. When there's oxygen, it can go through the Krebs cycle oxidative phosphorylation. But then if there's no oxygen, then it can become lactic acid, or it can become ethanol. Yeah. All right. Question 6. CMYC is a transcription factor that recognizes DNA bases using hydrophobic interactions. By binding to specific DNA sequences, CMYC can encourage cell division. In order to halt cell division, what mutations can one make to CMYC? Okay, so the essential thing to realize here is hydrophobic interactions. Because I see, like, GVV, LIA, GCA, TTG, I'm seeing that either we're having amino acids or nucleotides. So maybe these transcription factors are like, I don't know, like isoleucine, which is a very common non-polar and hydrophobic amino acid, and instead these are going to be hydrophilic and polar. So let's say you don't know any of these. What can you do right off the bat? Well, I can already eliminate C and D. Here's why. I can see that AFF-LLP and then FAFILL or IIL, these are very similar. So I can kind of assume and like predict that these are both going to have the same like high like polarity and thus like hydrophobicity. So therefore, because they're both so similar, these are not going to be the answers. So C and D will not be the answers. And then at this point, you really need to know your amino acids and how they like function. So I am seeing some polar like amino acids in this picture. So I see phenylalanine, which I know for sure is polar. So I would go with E in this situation, but yeah, knowing the 20 amino acids and their polarity, acidity, and whether they're hydrophilic or hydrophobic is pretty important. So yeah, the answer in this case would be E. But you can to make an educated guess, you can kind of eliminate C and D. And then CGTAAC. This one is kind of hard to like know because you need to know the codon wheel. Or sorry, like not the codon wheel, but the codon wheel. You need to know that which base pairs lead to which amino acid. But I already identified some of these as being polar, and therefore they would be hydrophilic. Yeah. So let's clear this. All right, number 7, a replication inhibitor is added to stem cells in the middle of DNA replication. At what phase of the cell cycle would the cells remain? All right, so I've run over this before about the mitotic phases, and we already know that that DNA replicates during the S phase. So this was kind of a gimme question. We know it's going to be D. All right. Ooh, this one looks a little tricky. So we have the chemical structure shown is AZT, also known as zidovudine, which is often used to treat human immunodeficiency virus or HIV. Based on the chemical structure, how would it work? All right. Let's see. I'm going to look at some of the functional groups. Right off the bat, I noticed this. This is probably the most important thing you can see. This is actually a nucleotide. This is thymine. Right there. So let me just draw an arrow to it. That's thymine. And then I'm seeing like a hydroxyl group. We have this 5-carbon ring. So already this is going to be something related to DNA. I haven't really read the options, but I know something's going to be about DNA. Maybe it could like replace DNA. So HIV, it uses reverse transcriptase. So instead of going from DNA to RNA like we do, it goes from RNA to DNA. So maybe it would just put like AZT puts this, or like it gets inserted into that like reverse transcription process, and then it inhibits the enzyme, or sorry, inhibits the entire process. Whoops. So let's go to the option choices. By decreasing the amount of DNA synthesized by HIV. I mean, because this could inhibit the reverse transcription process, this could be correct. I'll just put like a question mark next to it. It does seem pretty reasonable though. By modifying the surface proteins, no. By decreasing the fidelity of reverse transcriptase of HIV. I'm not even sure what that means. I'll actually search this up. What does fidelity mean? Let me search it up. But even though you're not allowed to do this on the exam, I just need to make sure that I am not giving you the wrong answer. The fidelity meaning. So fidelity, what is it? Wait, did I spell it correctly? Faithful or loyal. Oh, I see what it's trying to say. By decreasing like reverse transcriptase binding to HIV. That was kind of like a weird way of saying it, like the fidelity. Could have just said it like reducing how it binds, but like or something else. But it's not C, because we know this thymine, it definitely plays a role, and that's going to be in the like that reverse transcription process. D relates to proteins, not DNA, by accelerating and degrading viral DNA synthesized by the host. That doesn't seem correct either, because this option A just seems the most reasonable, because it would like insert itself. I'm not really like 100% sure about this, but that just seems like what most drugs could do. So yeah, I would say the answer is A. Whoops. Sorry about that. Okay, number 9. You have been asked to identify a bacterial culture using a light microscope in order to examine a single bacterium, you use the technique of oil immersion. Okay, I have very little experience. I've read a little bit about it, but if you guys know a little bit more about oil immersion than I do, I think you can write it in like the comments. So yeah, that could be very helpful. Because I'm not very experienced with oil immersion, but which of the following responses does not support your use of this technique? The numerical aperture of the objective lens is increased. Okay, yeah, this is a given. About about oil immersion, it protects the objective lens from contacting the slide. I don't think that's what it does. Like right now, actually, I would put this one as the X, because this was the one. It just doesn't really make sense. That's not really the purpose of oil immersion. Resolution is increased. Yes. The refractive index of glass is similar to that of oil, preventing loss of light to the scope. Yes. They both are around like 1.5. I think oil is 1.47 and glass is 1.5. It's not something you really need to know, but it is something that I just remembered from studying. So this is correct. So B seems pretty reasonable right now, but let's look at E. Refraction at the cover slip air interface causes the light ray to miss the objective. Well, that just doesn't seem to be the correct answer. Honestly, I am pretty much guessing at this point. But just my intuition is with B. If you do know why the answer is not E, you can write it in the comments, but E just doesn't seem to make sense. Like, air interface causes light ray to miss the objective is something that is quite reasonable. Like, it's definitely a valid point to make. Something like protecting the objective lens from contacting the slide is not something that I'm really aware of. especially with oil immersion. But yeah, write in the comments if you know more about this topic. But yeah, right now I'm going to go with B as the answer. All right, another question about meiosis. Which of the following is false about meiosis II? All right, so the cytoplasmic division occurs. That is true. That occurs in telophase. 2. Individual chromosomes line up at the equatorial plate. Yep, that's during metaphase. Sister chromatids separate and go towards opposite poles. That's also correct. That occurs during anaphase 2. The chromosomes replicate. No, that is not correct. I know that 100% is not correct. Because the chromosomes have already replicated in meiosis I. So no, this is definitely incorrect. So the answer, the answer is this. And then the nuclear envelope disintegrates. Even if you don't know that answer, you can already know that the answer is D. And the nuclear envelope definitely does have to disintegrate, because after meiosis I the cells are made, that nuclear envelope is going to have to disintegrate again so that meiosis II can occur. So yeah, the answer is D. But one strategy that I've come across with these questions, and that's kind of like a side tangent, but know that some of these options are a little bit above, could be a little bit above like your level of understanding. They definitely were for me. Like, I didn't understand some of the answers. But whenever I saw something that was blatantly incorrect or blatantly correct, then I just had to go with that one, because that was the one that just made the most sense, like in the context of the question. So yeah, 10 is D. All right, 11. Spores and seeds are necessary for reproduction. Which of the following is false with respect to spores and seeds? Okay. Now, as I skim through, I'm already seeing there's a lot of repeat within the answers. Like, you have a semicolon and there's 2 parts of the answer, and some parts are just completely repeated. So that could provide some hints, or it could just increase confusion. But let's see. Spores contain gametophytic tissue. Yes. Or wait a minute. Yes. That is correct. This is kind of touching on the alternation of generations. So, I always remember it as being the opposite. So sporophytes are going to make gametes. They're not going to produce spores. They're going to produce gametes. And then as I read that, my eye just came across D, spores produce sporophytes. No, that is incorrect. So the answer is already D. But you can just go through process of elimination, because you see that, look, spores produce sporophytes, and then spores produce mature gametophytes. So automatically, that A, C, and E are not the answers, because B and D are both conflicting. So it's going to be one of them. And yes, I remember that opposition, that spores, gametophytes make spores. Or wait, am I saying something incorrect? I don't know. But yeah, because I remember that opposition, spores produce sporophytes, the answer would be D. So it's important to remember the alternation of generations in the USABO. But all right, let's go to the next question. And this seems to be pretty similar. So if one were to compare the dispersal mechanisms of spores and seeds, which of the following statements would be false? Seed plants do not disperse spores. Well, that's clearly correct. Sorry. Yeah, that is definitely correct. Non-seed bearing plants are a paraphyletic group. Now, I don't remember my evolution so I'm just going to put a question mark next to this one. But I do think that this is correct. All right, so seed plants produce spores that develop into sporophytes. Oh, well, as I was mentioning, this is the opposition thing again. I think this would be gametophyte, not sporophyte. So, yep, C is automatically correct. alternation of generation seems to be coming up a lot more than I thought on this test, but okay. Let's go to the next question. This probably will be the last one for today. Number 13, monocots and dicots are angiosperms. There are approximately 200,000 species of dicots on the Earth today, and approximately 59,000 monocot species. From the characteristics below, select the statement that is true for dicots. So dicots are a paraphyletic group. I guess. So I do remember when I was reading Campbell's, there was polyphyletic, monophyletic, and paraphyletic, but I'm just completely blanking on that. I'm going to put a question mark. It could be correct, but I'm not 100% sure. Based on this information, how it's saying, like, maybe, but I'm not even sure. I don't remember. Having dicots have a ring shaped vascular bundle, but no pith region. That's incorrect. They do have a pith region. So yeah. B is not correct. They have a stomata in their roots. I don't think that dicots have stomata in the roots. The stomata is in the leaves. So you can have gas exchange, transpiration and stuff like that. So definitely not C. Have the order of plant components from medial to lateral direction in the root cross section of phloem, xylem to endodermis. Nope, that's not correct, because what I remember based on Campbell's is that you have like the phloem, I think, like this, and then you have like xylem like pockets in like a ring. So it's not exactly like that, because even though it's like phloem, xylem, it's kind of like this within the phloem. It's not kind of like distinct layers. So D just doesn't seem correct. And then E includes the family Rosaceae, Orchidae, and Asteraceae. Well, I already know orchids are monocots, so that's definitely incorrect. So, okay, by process of elimination, we can conclude that dicots are a paraphyletic group. So from this, I think clearly I need to review these paraphyletic, monophyletic, and polyphyletic groups, like that evolutionary history stuff and like making cladograms. But other than that, yeah, that was questions 1 to 13 on the USABO Open Exam. If you guys have any questions or know a little bit more, like another like solution behind any of the questions, please write in the comments below. Always willing to learn, and I'm really hoping to expand on my biological knowledge. Yeah, that's gonna be it for today. So yeah, next video I'm gonna probably post tomorrow, which I'm gonna do questions 14 to 25, and then as I mentioned, sets of 12 to 13 questions. And yeah, thanks for watching, everyone, and I will see you tomorrow.